Hello, my name is Seth Gammon. I'm the in vivo product manager here at Carestream Molecular Imaging, and today I'm going to talk a bit about how to utilize antibodies for in vivo molecular imaging. Many of us are familiar with utilizing antibodies for a variety of techniques in classical molecular biology. They've proven to be exceptionally useful in assays such as Western blots, where you then utilize a secondary antibody with a horseradish peroxidase or something like that attached to it. You can utilize antibodies for ELISA assays, immunohistochemistry, immunofluorescence, where either the primary or secondary antibody is labeled with a fluorescent probe, or FACS analysis, where primarily you're looking at fluorescently labeled primary antibodies. A digital imaging system, such as our Imaging 4000 pros, for looking at protein expressions via Western blot. One of the nice things about digital imaging versus classical film is that you don't have to worry about saturation. The software and camera will alert you if a pixel has actually become saturated. So you can much more reliably quantify changes in density. We also provide very simple tools in our molecular imaging software for conducting ratiometric analysis, ratioing one band to another. And with full 16-bit dynamic range, you can acquire both bright and dim bands in the same image. Many of these same principles apply when you go over to fluorescent Western blots. With the Pro, you can utilize readily up to 3 fluorescent reporters for imaging, interested in imaging 3 antibodies. You could utilize your first 2 colors for looking at changes in protein levels or phosphorylation of those proteins, and then that third color, which would have to be imaged in the green, for a very bright, robust London protein like actin or tubulin. These are normalizers that have to be present in really any Western blot to show normal loading or to normalize for loading of the different bands. The nice thing about conducting these experiments all in one, of course, is that you don't have to worry about stripping and reprobing and how that may affect your ability to detect bands after each strip and reprobe in the process. So the real question really comes down to Western blots and fluorescent Western blots are everywhere in vitro molecular biology. Can we, all of these antibodies are already out there to do in vivo Western blots, or are there specific applications in vivo where antibodies are particularly useful? Some of these other areas, besides Westerns, that would be useful in vivo are to develop therapeutic antibodies and to track where these therapeutic antibodies or antibody mimetics go, as well as to develop antibodies or antibody mimetics as imaging agents themselves. How all of these systems would work is that you would inject a either fluorescent or radiotracer labeled probe throughout the animal. After a long circulation time, it will clear away from the rest of the body and stay bound at the target site. Those clearance rates are actually a key variable that one has to understand when trying to set up antibodies. The next question one needs to think about is, what are appropriate protein targets for imaging antibodies in vivo? In lysase or in immunofluorescence, you've pretty much disrupted the cell membrane, and lysase is completely disrupted, and for immunohistochemistry, immunofluorescence, you've often permeabilized the cells. So antibodies can get through the membrane. However, for in vivo, or at least if you want to keep your animal alive and keep it in an in vivo experiment, you have to keep the integrity of your membranes intact. Thus, you're really primarily looking at targets that are expressed on the cell surface or in the extracellular matrix, relatively tightly bound to the cell. Imaging of intracellular ligands is really best left to small molecules or things like small molecules. equal these antibodies once we've selected an appropriate target, like an extracellular receptor. And then what are the pros and cons of utilizing those different labels? Well, one can utilize HRP for imaging lectins, we all know that. However, when you go in vivo, again, you don't want to inject high doses of hydrogen peroxide into your animal if you want to keep it in a vivo experiment, so that's really out. One can utilize visible fluorophores, particularly for high abundance proteins, as I mentioned, actin or tubulin. These are also fine for FACS or immunofluorescence, but really for in vivo, they aren't optimal and really shouldn't be tried fundamentally because you have such significant autofluorescence and limited tissue penetration of both your excitation and emission light. If you label your antibodies with near-infrared fluorescent fluorophores, these are appropriate really all the way across the board. This is where you want to work in the in vivo range because, as you know, your excitation and emission light and the relative amount of autofluorescence found in tissues in this region decreases significantly as well. When you look at radiotracers, some common labels are I-125, which is somewhat appropriate for westerns and a little bit for in vivo as long as you're working with small animals like mice and you have a very good radiosynthesis lab so you don't have significant free iodine running around. Indium-111 is very common, and also if you're looking at MRI, magnetic nanocrystals. I haven't listed any PET probes up here for a very specific reason, and that is, as I talked about, antibodies have to go throughout the body, circulate through the system, and then clear. This is actually a challenge for PET probes because antibodies have very long circulation times. Antibodies typically need, if you're using a radiotracer, these radiotracers have a specific half-life. Thus, by the time the antibody has cleared away from the surrounding tissues, you need sufficient amount of radiotracer bound at the target site and still decaying and thus emitting energy to be detected in order to see it. So probes like F-18 that have very short half-lives are just not appropriate and you have to wait 48 hours plus for it to clear. It's also been reported that it's not necessarily as easy to handle radioisotopes with antibodies. You have to have the appropriate regulatory and labeling facilities in your lab with shielding. Some people have that, in which case this really isn't a challenge at all. Another issue that's very common in selecting a target is the blood-brain barrier. The blood-brain barrier embodies out of the brain. So those targets, unless you're in a neurodegenerative disease state that disrupts the blood-brain barrier, are fairly well off limits. And finally, there is a pro and a con with having very high affinity antibodies. While it's a pro that your antibody binds very specifically and tightly to the target site, this could be a problem if you want to re-image your in vivo probe at multiple time points. So, for example, I showed the antibody clearing away from the non-target tissues and staying bound to the target tissue. Well, that target tissue is now bright or positive in whatever imaging modality you want to utilize. If you want to re-image at, say, 24 hours later, in order to have a very clear and decisive experiment, you really want all of the either fluorescence or radioisotope to be cleared away from that target before you start imaging again. And because of their high affinity, you really just have to test this empirically to see what the off rate will be from your target site and see what the time resolution, the potential time resolution of your imaging experiments might be. However, this could very well be an advantage for screening antibodies, whether they're therapeutic or imaging antibodies in vivo, because your bound signal isn't as time-dependent as peptides or small molecules. Thus, as you'll see in the protocol later, if you're off on your imaging protocol by a couple of minutes either way, it really won't matter because the antibody is bound very tightly to your target site. Many of these challenges have been addressed in the literature and by methods that I'll show you. One is if you, don't have the facilities to utilize radiotracers, and two, again, you don't have access to near-infrared fluorophores offer sort of an ideal solution. One, because antibody labeling kits are readily commercially available from multiple vendors, and you can quite easily add several fluorophores onto each antibody. The other nice thing about fluorescence is there's no real analog to radioactive half-life. Certainly, if there's some enzyme in the body that's chemically degrading your fluorophore or degrading the antibody and therefore degrading the fluorophore, there is a half-life associated. But the lights essentially aren't going out on a fluorophore in vivo unless it's being chemically degraded. Another solution is to make antibodies smaller. This has been nicely highlighted by the work of Dr. Anna Wu. Here, rather than having very large antibodies with long circulation time, you create minibodies or diabodies that are smaller. In addition, there are two-stage labeling processes where you inject either an antibody or a smaller minibody or diabody that has been labeled with, for example, biotin or some other affinity ligand. Thus, you can wait around for that to clear away from the body. There's no radiotracer attached to it to decay. You then chase it with a radio-labeled streptavidin. It will then find the site and hopefully clear faster than an antibody. However, this two-stage labeling processes are very tricky kinetics because you have two probes you have to optimize the uptake and clearance profile for. So we won't really discuss those any further in this presentation. So here are some examples I want to show you of successfully utilizing antibodies for in vivo imaging. Here's a PET tracer I-124 with a very long half-life. Each half-life is around 100 hours. So if you go by the rule of needing at least 6 half-lives, 600 hours is plenty for this, for even an irregular antibody to go around the body, circulate, and clear. Because you're using antibodies, it's facilely labeled tyrosine. However, again, you always have to worry a bit about deiodination. And these are cleared typically to the liver, kidneys, and bladder, as you can see here and here. The other nice thing about utilizing I-124 is that it's a PET probe, so it can be directly translated into clinical PET scanners without having to change labels, as one might have to do with a near-infrared fluorophore. Indeed, one would have to change out for some other sort of nuclear medicine imaging probe. These types of applications are ideal for the Albira PET, SPECT, and CT system. Using our in vivo imaging systems for 2D radiography, this group was interested in looking at antibody mimetics, so smaller than antibodies, and labeling them with I-125, injecting them, and then trying to see if they can target a tumor site appropriately. What they found is in their standard formulation, the tumor and liver were barely discernible from each other by 24 hours. However, when they made very simple changes to the formulation, such as adding penetratin or tap peptide to the injectable formulation, not as conjugates, but just adding it in solution, they were able to completely change the distribution of their antibody. And what's nice is that this was all done by 2D radiography. You don't have to go into a SPECT system until you get this nice, perfect formulation. They could then follow this protocol up with an imaging system, imaging section in the Albira system, for example, and actually look at the 3D distribution of these tumors and create that beautiful publication quality image. The other word of caution to take from this is that one has to be very careful about what you put into the formulation of your antibodies. You can see it can radically change the biodistribution of your imaging agent. So now I've talked a little bit about radio-labeling antibodies and antibody mimetics. Let's switch to fluorescence imaging. So it can utilize near-infrared fluorescence to track potential antibody imaging biomarkers and therapeutic antibodies in vivo. A couple of cautionary statements or things you need to make sure and do when you're conducting these experiments. is while you can certainly add as many fluorophores as possible, this could significantly affect the distribution of your antibodies, and if the fluorophores start collecting near the active binding site of your antibody, could actually affect its ability to bind to the target. Do purify your antibody away from unconjugated fluorophore prior to injection into the mouse. This will make sure that what you're imaging is really pure antibody fluorophore conjugate, because the distribution of your fluorophore will be completely different. And do, as with any in vivo imaging experiment, utilize multiple doses and delivery methods at first to find the optimal route. For example, check different doses of intravenous injections, as well as potentially different doses of intraperitoneal injections. This example of trying to develop a biomarker for looking at who was the director at Bristol Myers Squibb. They utilized an antibody that was specific for a cell surface receptor on EMT6 tumors and labeled this with Psi 5.5 as an infrared fluorophore. As you can see, by 4 hours, it's beginning to target the tumor specifically, but still has significant liver uptake. And indeed, one has to wait many days in order for the tumor to really be significantly distinct from the liver. So again, this is the big advantage of a fluorophore versus a radiotracer, in that you don't have to match it to the half-life. This fluorophore is fundamentally just on all of the time. This group also did some really nice controls. They did some nice controls where they just a random antibody that has nothing to do with their tumor. They can see that it clears nicely into the liver. So we knew that that tumor is not site isn't non-physically taking it up. And if you just look at Psi 5.5 as a dye by itself, it's also just a free dye, clears out quite rapidly. They then confirmed the liver targeting or liver clearance route through ex vivo imaging, which is always a good step after any imaging study. Another group at Abraxas was interested not looking at developing biomarkers, but rather confirming whether their therapeutic antibodies were selectively targeting a known site in vivo. These antibodies were labeled with Alexa 680 from Invitrogen's antibody labeling kits. 20 were injected into the mouse, and then mice were anesthetized with an injectable anesthetic. Mice were imaged in X-ray and white light reflectance imaging for anatomical co-registration, and also were imaged via spectral unmixing to help remove any residual near-infrared autofluorescence. What you can see from some initial studies is that the Alexa 680 has two nice foci, or the conjugate has two nice foci of uptake. However, without co-registration, we don't know what these are. When you co-register with an image, you can see that unfortunately these look like they're the kidneys. Then when you co-register with an X-ray image, one can clearly see that this is being taken up in the kidneys as opposed to the tumor. So unfortunately, this particular antibody was not appropriate for a therapeutic antibody, as it primarily lodged itself in the kidneys. However, when one looked at a different antibody conjugate, you can clearly see that there was very nice uptake and binding into the tumors that was specific. And again, the other nice thing about this protocol is that because of the long sort of circulation and clearance of these antibodies, you're looking at 24 hours post-injection. And because of the very tight binding to the tumor target site, if you're off by 5 or 10 minutes and just looking at this image, you're not going to miss this tumor binding. One can also readily quantify the fluorescence intensity and then ratio it, which is always a way to look at fluorescence intensity, to, in this case, the chest area as a non-specific target, will automatically calculate this ratio and average over all the tumors, and you get a value of 4.4 plus or minus 0.63. And the chance that this was just randomly different from 1 is less than 0.003. So in one simple experiment, they were nicely able to show that you have nice tight binding and statistically significant binding into this tumor class with this particular potential antibody. Indeed, you can also see a secondary site here in the fluorescence image. And that's because the system offers a nice wide dynamic range. And you can really see it much better in the spectral and mixing image, where you have a red spot just behind the initial primary tumor. The minimal autofluorescence that was actually in the previous images is displayed in green, and then the target site here is displayed in red. Both of these can be readily co-registered with X-ray in the multispectral software. One could think about using this multispectral capability not just for removing autofluorescence, because as we saw it wasn't a big problem in this particular experiment, but rather it could be utilized for screening multiple antibody fluorophore conjugates simultaneously for appropriate binding to either a tumor or a target site. So in conclusion, antibodies can be used for in vivo research, particularly when you label them with near-infrared fluorophores or with PET or SPECT labels when you match the half-life to the clearance time of your antibody. NIR imaging of antibodies is ideal for screening the potential of imaging bell sites such as a tumor or a localized muscle tissue. You can also utilize them for screening how well potential therapeutic antibodies or antibody mimetics are binding to the appropriate tissue, or are they binding to tissues you don't want them to, for example, like the kidneys or the liver. And indeed, by having a system like the in vivo MSFX Pro and the Albira system for looking at PET, SPECT, and CT, you can actually cover all of the possible antibody imaging techniques in vivo. So I'd like to thank you very much for your attention, and I'll be glad to take any questions.